and gentlemen, and thank you, the organizers, that I have the chance to present my case here. Atriva, we see that as a real new thing. This is the real new thing, a quantum leap in acute antiviral therapy. And why is that? This is my summary, in order not to bore you too long. Our scientists and technologists were the first one pioneering in platform technology which is targeting the host and not viral structures. And this gives us a lot of advantages compared to commonalities, common other drugs. And this mode of action is a very universal one and holds true for quite a lot of RNA viruses causing severe viral diseases. Meanwhile, we are a clinical stage company. We are entering phase 1 development within the next 4 weeks. First patient will be dosed, first volunteer, and would like to now to develop our our lead compound, ATR02, to a sensible clinical endpoint with 2 phase 2 validated studies by the next 3.5 years. We have collected an experienced management team, and these are advised by internationally renowned consultants and advisors. Our lead compound, ATR02, our indication is influenza for the elderly and the compromised people. This is a kinase inhibitor, a MEK1 inhibitor, very target-specific, not ATP competitive. And by using those technology, this is, as I said, a commonality for various respiratory diseases, viruses, including RSV, including coronavirus, including metapneumo, but first and foremost against flu, influenza. This is, as I said, our lead indication. The advantages we could be demonstrating preclinical research and development is that we don't see any resistance development, also against emergent strains. We have a much broader therapeutic window compared to standard of care. And we got also the advantage that we can guide our clinical development with a developed biomarker, which we developed in our labs. Our first and foremost indication is influenza, and more than ever, it's a therapeutic challenge. We had a record season last year. This year is a little bit less, luckily enough. The flu shot is better than last year, and this was the issue of last year, vaccine mismatch. The efficacy of the vaccination was poor. And in addition, also our antiviral drugs we got currently are not as efficacious as you would like to see it. And there's always the resistance of the emergence of resistant viruses. For example, with the newest entrant, they did see it already with up to 20% in clinical development. And what you don't have up to now licensed is a treatment for people at risk developing flu-related complications like pneumonia. And if a new virus comes in, a pandemic one, there is nothing. Just nothing, you can just pray. What is our solution on that? We are targeting host cell factors that block the RNA virus replication. And we know that all of those viruses are depending on the intercellular RAS-RAF-MEK-ERK pathway, which is leading then to risk and then to the kinase. And this is a phosphorylation event which the virus is needing to replicate itself. All of those viruses don't have the ability to replicate outside a cell. They need a cellular growth machine, which is triggered by phosphorylation events, which is the actual step of activity to collect and to assemble viral proteins and viral. And with our ATR02, we can effectively block this step and can suppress viral replication. What we could show with our lead compound, we got a broad efficacy against new and old influenza strain, every emergent strain which is reported to us, and we have a very broad access via one of our founding professors in the University of Marburg-Gießen, which is a German mainstay in infectious disease. We don't expect any resistance development we could even show that after multiple rounds of virus propagation. of course, we have an efficacy against Tamiflu, and in the meanwhile also against baloxavir-resistant strains. And we got this broad therapeutic window, and we see an efficacy in vitro and in animals of a variety of other respiratory viruses, including RSV, metapneumo, hantavirus, and corona. And this is just a data set we got here. Here you see an experiment in mice where they get a lethal dose. Mouse is the common in vivo setup to prove the efficacy of antiviral, anti-influenza drugs. We are seeing here, we are infecting here mice with a lethal dose, 5-fold lethal dose of influenza. You see all of the non-treated mice are dying, and really all of them. And even after 72 hours after infection, there is no other drug around which is really demonstrate that besides the one of Vertex. We still see a clear, significant activity of mice receiving our treatment, and this is certainly a very important proof of concept of our technology, which led us also to develop the drug. In the meanwhile, we have finalized our preclinical development, the full package. Chemistry, manufacturing control, full tox, 28 days in 2 species. We did not see any showstopper, no liability. We are starting our first clinical development in March 2019, our phase 1 study. This now leads me, what is now the clinical target product profile of our lead indication? I mentioned already it's a small molecule, a non-competitive ATP kinase inhibitor. Non-competitive means no competition to ATP, and so also no off-target and side effects. Our target group are patients at risk, the elderly, and those with certain concomitant diseases like COPD, asthma, heart disease. And the target dose is most probably 300 milligram. We will see that in the next year in our early phase clinical development. And what we can predict and can see out of our preclinical development file that we do not see any liabilities, and we don't expect any side effects because of the short treatment regime which we expect. By the way, it's an oral treatment, tablet once daily, a stable formulation which can be easily converted to a marketable formulation. And this has already appreciated in the first contact with the authorities. This is our clinical program which we are running in the next 2 to 4 years, starting with a phase 1 study, followed by the phase 2A challenge study. Challenge means we are infecting healthy volunteers with a topical influenza strain, a real active one out of the flu seasons 2014, which is identical to the one we have currently. And this is a study in 3 arms and placebos, where we as well looking into our claim with a prolonged treatment window. So we will have 1 arm here, there, only after 72 hours the volunteer will be treated after infection, which is the maximum which the ethical committees are allowing us. This is followed then by a classical phase 2B study, and then by a phase 3 study. And these are the 2 value inflection points also for any type of investors and investment joining us. And it's very clear, and we got previous experience, the team is experienced in running these influenza studies. We did it beforehand and are expecting here no surprises. The only surprise would be if we wouldn't have a flu season in 2021 and 22, which I don't believe, unfortunately, for the world, fortunately for us. Influenza, I feel there is still no big deal in discussing the huge market opportunity here. And it's unfortunately, since 50 years of research, there is not really something coming up short to mid-term. And especially as all classical approaches coming up right now do have this resistance issue. And this is why we feel it's really worth investing here, time, effort, and money. I mentioned already that this is a universal platform type approach here, and this is why we have been building up a pipeline. Our front runner is influenza, but we have also started a program on hantavirus. We will be filing a US-American orphan drug designation. This is still the old plan, OMPD, but we go to the United States. We have just decided that because there's much more cases there of Hanta. In the Sierra Nevada, in the Rocky Mountains, you find them. They infect people and they're dying from them. It is followed by a program on RSV and COPD. And we have also a drug discovery program started and last year, which we are hoping to get new compounds out, especially in the risk inhibitor arena by mid-2020 for preclinical development. So our goal is really to building up a pipeline here, a platform. Having said that, this is the financial need we have in order to get there, what we expect. We are now here. This was the seed round. We have now collected the initial 25% of our running Series A. And if we close that, we hope to have that done by early April this year. Then we'll be having the funds to finance the first 2 clinical studies plus our pipeline, and follow that by the next round, again on 15 million for the series, first of the phase 2 study, phase 2B studies. So in total for the next 3 years, 30 million minimum in order to bring it to a full proof of concept. We are backed by a broad and dense intellectual property family, net of intellectual property and family. These as a mixture of use patents, combination patents, also in the meanwhile composition of matter patents of our newest compound, which gives us exclusivity up to the year 2038. And this is run by an experienced team with all our managers. We did it successfully beforehand. Christian Ballasch was COO of Intercell, built up there, installed there the project management function later on, and is a serial entrepreneur. He did his PhD in the labs of Axel Ulrich. Sebastian Canisius is very experienced in running influenza and other respiratory virus studies. Did it before. Oliver is the inventor of the technology and serves as our CSO. And I am just doing, since 30 years around, and doing these things. So, and in summary, so we got here a unique chance with a real new approach which solves a lot of problems that current influenza drugs have, with an upside for other viruses. Our lead compound enters clinical development. We have 2 value inflection points in the next 3.5 years, and we are notorious to keep our timelines. We did it up to now. Once we got the funding, we'll continue doing so. And as I said, we are currently ramping up a venture round. We had a first closing earlier this year, and would be very happy to entertain all questions on that. And this is the team. We are currently 10 people.